The nucleic acids will be the fourth and final kind of biological macromolecules that we will be discussing. Like carbohydrates and proteins, nucleic acids are polymers made up of monomers known as nucleotides. Nucleotides are actually composed of three main things. Okay, so each of the monomer of a nucleic acid is composed of three things. First will be a phosphate group, then there's a sugar, and then there's a base. Now, depending on the type of nucleic acid, these two types of nucleotides as shown here may be present. In RNA or ribonucleic acid, the nucleotide is known as ribonucleotide. And the name is actually derived from the presence of a sugar known as ribose. In DNA, the nucleotide contains a sugar known as deoxyribose. Now, deoxyribose differs from that of RNA's sugar because of the absence of one oxygen atom that's attached here on the second carbon of the sugar. Nevertheless, both of these nucleotides have those three parts that we mentioned a while ago. Again, a phosphate group, a sugar, and a base. Then we have something what we call as nucleosides. Okay? Nucleosides would be similar to a nucleotide in structure, as you see in these two, but there is no phosphate group attached. So only the sugar and the base. And then we call that a nucleoside. For each of these two types of nucleic acids, RNA and DNA, 4 types of nucleotides may be used to build up the polymer or the nucleic acid. So those 4 types of nucleotides for RNA would be a phosphate group, a sugar, and then a base. The base could be either adenine, the one shown that's attached here with the letter A, or it can be guanine. So adenine and guanine are purines, and their structure would be composed of 2 rings, as we see here. Or it can be cytosine or uracil. So cytosine and uracil are pyrimidines, and their structure is made of only 1 ring of carbon atoms. For DNA, the nitrogenous bases that are attached to the phosphate and the sugar are exactly similar to those of RNAs, except for one of the pyrimidines, and that's thymine. So instead of uracil being attached or partnered to adenine, in DNA we have the base thymine. So you might imagine already that nucleic acids are polymers, and then these polymers are made up of 4 different types of monomers that are attached randomly onto each other. If you compare this with the story of proteins, remember there are 20 amino acids that would comprise a protein polypeptide. Okay, that's the potential. Versus nucleic acids, as we see here, if we consider DNA, there are only 4 types of monomers. There was once a point in science when scientists were actually looking for the molecule that carries the genetic information in cells. And their final 2 contestants for that molecule were the proteins, because there are 20 types of amino acids or alphabets that can make a polypeptide, and DNA, a nucleic acid, which has 4 types of nucleotides that can make the polymer in DNA. So eventually, an experiment by Hershey and Chase actually showed that it's DNA that actually carries the genetic material in cells, while protein was relegated to something that's produced when the genetic material is actually read or expressed. So DNA might be the main carrier of genetic information in cells, but there are some sub-life forms, like viruses, that actually use RNA instead of DNA as a storage or carrier for their genetic information. So, how would nucleotides polymerize? In carbohydrates, in proteins, and even in lipids, we learned that a dehydration reaction would actually cause the monomers or the components of a fat like the glycerol and the fatty acid chain to actually bond together. Now, in nucleic acids, it's a little bit different than that. So, a free-floating monomer of a nucleic acid is actually attached to 3 phosphate groups, as we see in the bottom portion of the illustration here. Okay? So, what happens is, with the aid of an enzyme called a polymerase, the hydroxyl group on the 3' carbon of the sugar, as indicated by the arrow here, attacks the first phosphorus in that 3-phosphate chain on the free nucleotide. And what happens is a chain reaction so that the other 2 phosphate groups would detach from the nucleotide, and the nucleotide will now attach to the 3' end of a building nucleic acid. So this bond that is formed with the aid of an enzyme called a polymerase is called a phosphodiester linkage. And we notice here 2 ends of this particular strand. We have the 5' end on the upper portion, okay, because of the 5' carbon that's attached to the phosphate group. And then a 3' end here at the bottom, which actually contains the hydroxyl group of the sugar. Now, this alternating arrangement of phosphate sugar is considered as the phosphate sugar backbone of the nucleic acid. And take note that it's only in the 3' end of this chain where a new nucleotide may be added into the nucleic acid, again with the aid of an enzyme. In DNA particularly, the nitrogenous bases on the nucleotides will actually form hydrogen bonds and bond onto each other, like 2 sides when a zipper is closed. This bond is actually called a hydrogen bond. So a hydrogen bond runs along the center of a DNA molecule, attaching 2 complementary strands, as we see here. So these 2 strands are actually anti-parallel, and we see the bonding preferences. So for the green bases here, we have adenine and thymine, and for the orange bases here, we have guanine and cytosine. Notice that for guanine and cytosine, 3 hydrogen bonds are used, okay, as opposed to 2 between adenine and thymine. So DNA not only zips as a 2-stranded nucleic acid, but also twists on itself to form a shape we call now as double helix. RNA, on the other hand, remains strictly single-stranded. But it doesn't mean that it exists just as a straight line of a series of nucleic acids. In fact, as shown in this drawing, many RNA types like this, tRNA, so this is a tRNA, may actually fold onto itself. So it can fold onto itself so that some of its nucleotides are actually bonded to its own nucleotides on the other portions of the molecule. So the bonds here are also hydrogen bonds. So you have here a formation of a looped molecule for the tRNA. And sometimes the RNA molecule can be very long that multiple loops are made as the RNA wraps onto itself. So this is mostly observed in RNAs we know as ribosomal RNAs. Then I would like to include viruses in this video, not because I want to focus on discussing them, but instead we look at them now as sub-forms of life which are not really considered as living things, because they cannot reproduce their own when they are outside their host cells. But what we might want to observe in viruses is that they are made of a protein coat. So there's protein with their genetic information stored inside the protein coat, either as DNA or RNA. So here we have 2 viruses, the tobacco mosaic virus on the left and a bacteriophage here on the right. Viruses may actually self-assemble inside their host cells, okay? Because as they hijack a host cell, they inject their genetic material, and the genetic material somehow takes over on the instructions that are going to be read by the host cell. And the instructions will now shift to make more viruses. And as the virus proteins and nucleic acids assemble inside the cell will eventually lyse or break, and the new virus particles will break free. And in some chance, they might attach to other bacteria, okay, and other cells and infect them, and this cycle will repeat itself. So again, the concept here is self-assembly of certain types of molecules, especially in viruses, which the origins of which may be tied to how the first forms of life really came onto our planet. So this wraps up our discussion on the chemical basis of life.